So, while cells really are computers, just like, this computer here, they're different in that their programming was done randomly by, producing a whole large number of computers and destroying any that didn't work, and then over and over again. And in the same way, we have the production of viruses. Now, how viruses have been produced randomly? Well, even in a living cell, before you get viruses, you have what are called jumping genes. You have genes that encode enzymes that can then snip out that gene and then paste it somewhere else in the same genetic sequence, in the same genome. Okay? So these genes can jump around within the genome itself, right? They already encode machinery that allows them to infect the cell when they erode it, right? So we have genetic sequences that can hop around within one cell. To be able to jump from cell to cell, all you need is to be able to cut and paste yourself from one genome into another, like whenever the cells come into contact or whatever. And today we have viroids in plants, which are just naked nucleic acids that can infect plants and make them manufacture more of those viroids. The viroids are able to infect plant cells because plant cells are connected to each other, cytoplasm to cytoplasm, so the plasmid is product. And there's also these things called leaf hoppers, which will suck the plant juices. And as they hop around spreading that stuff, they spread these viruses, on their mouth parts, right? So naked, nucleic acid can work as a virus. In most of the systems, however, viruses have evolved to have something called a capsid. So true viruses have a nucleic acid core and a protein capsid. Okay? The protein capsid is a self-assembling set of repeating subunits that will make themselves into some sort of shape, either a cylindrical shape or a very complicated sort of shape, or even an icosahedron. To show how this works, this guy developed something like this in plastic on a much larger scale. So what he showed you here is that the shape can be disrupted by shaking it vigorously, and then by shaking it more gently, it can be reassembled back into its original shape. And you see that the pieces kind of bounce around and they self-assemble in the shape, and then the last piece really doesn't want to go in, so you just keep shaking it and shaking it. And then eventually it does snap back together. So this is an example of self-assembly. You can make things in such a way that they will self-assemble when given this random impulse of shaking. And the random impulse of shaking in you is heat energy. Heat energy causes Brownian motion, so these particles shake and stuff like that until they settle and sink into the most thermodynamically stable shape. And the most thermodynamically stable shape for these viruses is these different shapes, right? So like in many human viruses, like this one, and this one, the icosahedral or 20-sided base. And you can see how it's the same single piece that's repeated over and over and over again, and when it comes and fits together, it makes this 20-sided shape, right? And that protects the nucleic acid inside. And I say nucleic acid because some viruses are DNA viruses and some viruses are RNA viruses, right? So there's some variety in what kinds of random things you get. So here we see that there are different viral shapes. We have the very simple viruses here, like tobacco mosaic virus, has just an RNA, helix shape, like a corkscrew kind of shape, and it's covered with, the repeating subunits of the capsid. Here's an icosahedral one. Here's a bacterial one, a much more complicated capsid. It's still just a method to inject the DNA into the bacterial cell. So this is a pathogen of bacteria, this is a pathogen of plants. Adenovirus, influenza virus, they're both pathogens of people, right? These, in addition to the capsid, they have something else on the outside called a membrane. Many animal viruses have and require a membrane to function. The membrane is membrane taken from their last host. That membrane, in part, it helps them infect animal cells because animal cells are covered by a membrane, so it makes it much easier to sneak by the immune system and everything that you're wearing, the skin of your previous victim and so forth. But on the other hand, it gives the viruses a huge limitation in that they can't just like sit out on a surface for a long period of time, right? They have to be within chemical environments that are conducive to the maintenance of a cell membrane because they cannot maintain their own cell membrane, right? So, like the AIDS virus can't just like sit on a desk for a period of time. It needs to be in blood or some other living liquid that will help maintain that cell membrane because the AIDS virus can't do anything to maintain the human cell membrane. It's got to be outside of it, and without it, it cannot infect. So several animal cells are dependent on that, but not all. Rabies, for instance, doesn't have that. So anyway, there's different kinds of these. Now, viruses are obligate intracellular parasites. They have no metabolism on their own. They're not autotrophs or heterotrophs. They have nothing to do with food. No metabolism, no food or anything like that. So what sort of reactions, what sort of biological reactions do they engage in? They replicate themselves. And to replicate themselves, they need to be in a cell. Okay? So you can't replicate viruses just like in a flask, right? Like you can with bacteria or whatever. With bacteria, you can put them in a bottle, give them food, and they'll make more of themselves, right? Viruses, you can't do that. So if you want to culture viruses, you have to keep probably human cells right here being cultured in these flasks. That's what usually culture human cells. Another way is chicken eggs, right? You can cultivate viruses in a chicken egg, and the nice thing about like cultivating rabies or something like that in a chicken egg is that then it just sits there in one place and isn't going to run around, and no one, if you're cultivating a dangerous virus, you don't have to be like, wait, where did it go? I had it right here a second ago, and then it's gone, right? So you can just inject the different viruses into the egg and infect chicken eggs, even with viruses that chickens normally can't get, because if you're injecting it into the egg or whatever. So that's a very handy way to culture certain viruses. And so all different kinds of viruses have different culture requirements, but the basic idea is that it has to be done within a cell. Okay. I think we can talk about, we can move on to bacteria yet. Okay. Right, bacteria. So bacteria are, what are the three domains of life? Eukarya is one. Eukarya. What's that? Prokarya. There's two prokaryotic domains. Archaea is one, and bacteria is the other one. Okay, so it's bacteria, archaean, and eukarya. Those are the three domains of life. Right. Bacteria, like eukaryotes, they are prokaryotic. What does prokaryotic mean?
They do not have a nucleus.
They do not have a nucleus or any other membrane-bound organelles. Okay, so only eukaryotes have membrane-bound organelles. And by membrane-bound organelles, I mean anything where you have a cell membrane made into a bag inside. So the endoplasmic reticulum, the lysosome, the Golgi apparatus, any of that kind of stuff. You don't have any of those kinds of things in a prokaryotic organism. Okay, so let's look at the anatomy of the prokaryote. How do bacteria reproduce? Binary fission, right, by binary fission. So the cells will just divide into two. All right, so the most basic thing we have here, we have the nucleoid. Here's the bacterial chromosome in the center called the nucleoid. We've got the cytoplasm right here. And then we've got a layer of cell membrane around that, right? The cell membrane right here, this inner yellow layer. And that's the cell right there. Now, most bacteria have an external layer outside of that, okay? So here, rendered in a little darker pink, you have the cell wall, all right? So bacteria have a cell wall that has helped give them resistance against water flowing in the cell, so it gives them a little bit of osmotic resistance. And then outside of that, you may or may not have another layer called the glycocalyx capsule or slime layer outside of that. So in the cytoplasm, we have ribosomes as well as inclusion bodies, which are just droplets of like oil or crystals of sugar or whatever. And then we might have fimbriae, a fringe of little protrusions from the surface of the cell, which might be used as grappling hooks. We have pili, which are these hollow tubes that stick out of the bacterium, in which they inject DNA into another cell to genetically engineer it, right? So bacteria will pass on plasmids through these pili, right? And the plasmids are not part of the bacteria's chromosome, because the chromosome is part of that bacteria, what species it is. What they pass on is these genes that you could think of it in a lot of cases as like giving the recipient some kind of superpower. In a lot of cases, it would make the recipient, the bacteria might pass on another cell the ability to